And thank you to EMA for giving me the opportunity to present this project in front of this highly qualified audience. This is an example of about development of alternatives to antimicrobials supported by the European Union. And you can see some details about the project on this slide. I would like to remark it is a typical example of multi-actor project where not only researchers, but also professionals from different sectors are collaborating together to, in this case, develop alternatives to antimicrobials. And it is, as part of the consortium, there are 5 universities, 4 industries, 4 SMEs, and 1 large veterinary organization, which is the Federation of Veterinarians of Europe. So for a total of 14 partners from 9 countries. And I became familiar actually with this terminology, technology readiness level, with this project. It is something that is used about the development of commercial products and bringing products closer to the market, in particular innovation actions, which is the instrument, the financial instrument that is supporting this project are supposed to produce new or improved products and bring them, in this specific case, from TRL level 5 to TRL level 7, where 5 is basically testing the alternative in an animal facility, and 7 is testing a prototype of the alternative in an operational environment, which means a farm where pigs are sent to the slaughterhouse. And we decided, our strategies was to decide on a single animal species and a single disease. And the reason was that pigs account for the vast majority of antimicrobial consumption in the livestock sector. And here there are some data you can see in the graphic reporting the biomass production of different animal groups on the left, and on the right the amount of active compound of antibiotics in Denmark. You can see that although pigs represent less than half of the biomass, they account for approximately 80% of antimicrobial consumption. And most of these antibiotics are used during the weaning period. The weaning, for those who are not familiar with this term, is the time where the pig move from milk-based diet from the sow to solid feed. And this is an extremely stressful time for piglets in intensive production systems. You have to think that the natural, in nature, sows would wean normally their piglets around 14, 17 weeks. And in intensive production, this is reduced to 3, most countries 3 weeks, in some countries like the Netherlands, as much as I know, 4 weeks. So these animals are extremely fragile, and they have to face a stressful condition because of the change in the diet, but also a social stress because they are mixed together with piglets from other litters. And this is extremely stressful, and the stress results in a susceptibility to specific pathogens that cause diarrhea in these animals. And the most important of this pathogen is an Escherichia coli strain that is producing toxins, but this is less important in this context. What I would like to remark by this slide is that there is actually, this is a very good example where there is need for new antimicrobial solutions, because there are some vaccines that are available, but for a number of reasons, it is extremely complicated to develop an effective vaccination program due to the age of these animals, but also the complexity of the disease, the variety of enterotoxigenic E. coli that can be involved, but also there are studies showing that these enterotoxigenic E. coli are they're not the only pathogenic pathogen involved in the etiology of the disease. So there were two classes of antimicrobials that were extremely effective in controlling this disease. Zinc oxide, which has been banned to prevent the transmission of MRSA, methicillin-resistant Staphylococcus aureus, from livestock to humans. And colistin, that has been mentioned in the previous presentation, that has been restricted, especially in Denmark, but all over Europe, because of the risk of transmitting colistin resistance in gram-negative pathogens. And then there is, shown in the graphic on the right side, there has been an increase in resistance to the antibiotic that is used as a first choice for treatment of this disease, which is neomycin, an antibiotic that is actually very little used in human medicine, primarily as a topical product, and is used orally in pigs because it has a very low oral bioavailability, so it concentrates in the intestinal tract and is poorly absorbed, and therefore reach very high concentration at the site where enterotoxigenic E. coli proliferates. And as it is shown in the graphic, when neomycin was reintroduced in Denmark in 2017, the prevalence of resistance in clinical isolates of this pathogen was relatively low, around 10%. And after we have started using this drug, we have seen an exponential increase in resistance. And these are data from pathogenic strains isolated from diseased animals. And the last data from 2022 indicated that ETEC have reached about 40% resistance to neomycin. This data may be biased because not all veterinarians are sending samples to the diagnostic lab, but this roughly tells us that at least 1 out of 3 infections, outbreaks caused by ETEC is resistant to the first choice antimicrobial. So there is need for solutions here, and this is why we decided to focus on this disease. And even though we focus only on one single animal species and one single disease, we decided to have a very broad and comprehensive approach in terms of types of solutions. So the AVANT portfolio includes 4 categories of alternatives, the so-called gut microbiome modulators, which include symbiotics, a mix of probiotics and prebiotics, and a specific on-farm protocol for fecal microbiota transplantation. I will touch upon this in the next slides. Then some products that fall more under the category of veterinary medicinal products, in particular some polymers and mixes of phages that were that target this pathogen, enterotoxigenic E. coli. Immunostimulants, so drugs that act on the immune system of the animal, and we have tested both oral and systemic products. And finally, a 4th work package was dedicated to feeding strategies targeting either the sow or the piglets. So the aim of the project was to develop and test these alternatives and see what is their impact actually on antimicrobial use in pig production. And this is a little bit the overall structure of the project, showing the 4 scientific work packages where these products, these alternatives were developed and tested in preclinical studies. And now we are actually at the stage where we move from the animal facilities to the clinical trials that will be performed in 3 countries, including the Netherlands, Denmark, and France. And of course, based on the results of the preclinical studies, we made the selection. We could not test all the interventions, and for some of them, there were also legal constraints in using them in real commercial farms. So we did an evaluation of the results, and 2 interventions were selected based on their efficacy. One is fecal microbiota transplantation, which I will cover in detail during the next slides, developed by the group of Professor Thomas Thumann at the University of Copenhagen. And the second one was a fiber-rich diet of the piglets, developed by a small medium enterprise in the Netherlands, Scottish Feed Research. And as I said before, now we are in the most exciting phase of the project, where we test these interventions and not only assess their efficacy in preventing and controlling the disease, but also the cost and therefore the sustainability of the interventions and the effect on antibiotic use. So I will start from some preclinical data regarding fecal microbiota transplantation. And before presenting the data, the overview of the data, I should mention that this is a protocol that is performed on farm, meaning that the donor of the feces are recruited within the farm where the protocol is applied. So there is, in such a way, we avoid the risk of spreading pathogens from one farm to another. And initially, we tested 3 different types of fecal transplant. The total transplant, the gastric intestinal transplant, the gastric transplant, and also for the fecal transplant, we tried a filtrate to filter, because there is some evidence from human studies that filtered feces may actually have an equal or even better effect compared to intact feces. And I should also mention that fecal microbiota transplantation, for those who are not familiar with this procedure, is currently used in human hospitals for control of specific intestinal disorders, especially those associated with Clostridium difficile. So when we tested these 3 inocula in piglets, single-housed piglets, at the animal facility, we were not particularly excited about the results. You can see in this graph on the y-axis the percentage of animals displaying diarrhea, and on the x-axis the days of age. And as I said, the weaning starts at around 3 weeks, so day 21. And we could see a significant effect only for the fecal filtrate around day 28 and 29. This was statistically significant, but as you can see from the graph, we could still see around 20% of piglets displaying diarrhea at day 28. So it was a reduction in the number of diseased animals rather than a complete prevention of the disease. And this second graph is reflecting the same, is just expressing, instead of the number of piglets displaying diarrhea, the consistency of the feces in the 3 treatment groups in comparison with the control group. And again, we saw a significant difference at day 28, meaning that the feces were more solid in the piglets treated with the filtrate. So we decided to move on and test this protocol in an environment that is closer to a real farm, also because these piglets were artificially inoculated with the pathogen. I forgot to say that on day 24, as you can see on the first graph in the middle. So we moved to an experimental farm where post-weaning diarrhea occurred naturally. And the filtrate was administered during the first 6 days, so when the piglets were still together with the sow. And as you can see here, the weaning was implemented at 4 weeks, day 28. And periodically, there was a clinical and fecal score of individual animals that was performed throughout the study until the animals were euthanized at day 40. And in this case, the results were much more promising compared with those obtained under controlled conditions. So we saw that the daily occurrence of piglets displaying symptoms of diarrhea was significantly lower in the treatment group compared with the control group. And here, the number of asterisks are only indicating the level of statistical significance. But as you can see, throughout the post-weaning period, the fecal filtrate was able to reduce significantly the number or the percentage of piglets that were affected by the disease. And of secondary importance in the first days of life, so when the piglets were still treated with the filtrate at day 6, we could also see a lower percentage of piglets displaying neonatal diarrhea, which is the second most critical phase for antimicrobial use in pigs after weaning. And this graph is showing the effects of the treatment on mortality. Again, there was a significant reduction in the number of pigs that died or were euthanized because of the severe clinical conditions that they presented. This, because of the study design, antibiotics were not used during the study. So when the animals were had displayed the severe illness, they were euthanized. So there is very good news in this graph that by using this approach, which is relatively inexpensive, we can actually reduce from around 30 to 20% the mortality in piglets. This is good news, but at the same time, we have to realize that without using antibiotics, we would have to lose about 20% of the production at this stage. So then we had to move to the real farms. As I mentioned before, fecal microbiota transplantation was selected for the clinical trials. But 2 of the countries, Denmark and France, denied the authorization to perform the clinical trials because of public health safety. I have to mention that there are presently some regulatory constraints for the marketing of this product and even for testing the product in pigs that are supposed to go to the slaughterhouse. And this is because it is not a feed additive, and according to the current EU regulation, feces or any derivatives of feces cannot be used to feed animals. And is not obviously a medicine because its content is not standardized, and even the mode of action of this fecal filtrate is not elucidated. So I hope that this project will also help to start a process, a dialogue with the stakeholders. And I forgot to mention in the beginning that AVANT is a multi-actor project, not only because of the composition of the consortium, but also because we have the pleasure to have in our external advisory board different agencies, including EMA and EFSA, and different associations representing the stakeholders involved in this fight against AMR in livestock production, like COPA-COGECA representing the farmers, FIFAC representing the feed industry, and the animal health industry as well. So I hope that by this project, at least we will be able to set a path for future projects, so that it is known what can be done and what are the requirements for testing these products in farms. There are also some technical challenges that should not be overlooked. We haven't actually paid a lot of attention to the quality of the donors. We have just selected some healthy animals, but much more research can be done to select the optimal donor feces. We have seen that filtrates are more effective based on our data than the actual feces. But this requires some on-farm equipment like centrifuges and filters that are needed for the implementation of this protocol on-farm. And then finally, also handling the piglets individually is not particularly easy, and is also a little bit stressful for these animals, of course. Then I will mention a survey that we have conducted among the end users, or at least the 3 categories of end users of these products, pig farmers, pig veterinarians, and consumers, to understand the level of acceptance of different types of alternatives to antibiotics. And this was conducted in 2021. We subcontracted a specialized company, PNM, that is based in Germany, which conducted a telephone survey on a large sample of respondents belonging to these 3 categories. And this was conducted in 5 countries, which include some of the most important pig producing countries in Europe, and also culturally, we try to cover as much as possible the diversity within our continent by including countries from Northern, Southern, and Eastern Europe. And this is a long publication is on the way, and so you will soon get access to the full results. But if I should highlight the most interesting ones, is that all 3 categories, including farmers and veterinarians, have a better perception of the traditional measures like vaccination, biosecurity measures, compared to more innovative products like fecal microbiota transplantation and phage therapy, meaning that in addition to the regulatory challenges that we are facing, we are also facing some cultural challenges. So part of the work will also be to raise awareness about the efficacy of these products among the professionals, and maybe also to emphasize the safety of these products from both public and an animal health perspective. And then the second element that I have decided to highlight here is that about 70% of the consumers on average across the 5 countries said that they are willing to pay 10% or more to buy food that is not produced with animals treated with antibiotics. And here we have to be careful in judging this result or interpreting this result, because there are actually real data showing that consumers are not particularly attracted by food products that are more expensive, even though they are not produced with antibiotics. And a good example of this is that in Denmark, the Danish pig industry decided to start a production, an antibiotic-free production, some years ago. And this is actually going to be shut down next year. At least this is what I have heard, because it doesn't seem to be a profitable market. So some take-home messages and maybe some point for a discussion at the end of this session. First of all, let's be aware that based on the state of the art, we do not have alternatives that are able to completely replace antimicrobials. It seems that some of them may be useful in reducing the use of antibiotics, but we still need antibiotics for curing the diseased animals. And in this respect, it would be important. So far, at the European level, we have using this benchmarking approach, and we have just been told that the latest reduce the overall antimicrobial consumption by 50%. But if this is the situation, we need to understand when the reduction will actually start affecting animal welfare. The graphic on mortality was quite clear. If we didn't use antibiotics in that farm, 20% of the piglets would have died regardless of the alternative treatment. This is one point. The next one is that we have been funded to test by the European Union to test alternatives to antimicrobials, and according to the regulation of the European Union, we are not allowed to test them in clinical farms. So we need to align a little bit the regulation to the needs of this society. There is a big societal pressure to combat AMR and reduce antimicrobial use. So as I said before, I hope that, first of all, we will have this tested, the fecal microbiota transplantation in this country, in the Netherlands. So I hope that the results of this clinical trial will convince more countries to accept testing, more trials that will be, of course, needed before this approach can be marketed or at least legalized at the European level. And the last point is that, as you can see, there are also some cultural barriers that we have to face, not only among consumers, but also among veterinary professionals and farmers. And altogether, my point here is that we need a holistic approach that is involving political decisions and also public acceptance. And if we really want to avoid the routine use, metaphylactic use of antibiotics in specific production systems like the intensive pig production, this require also a change of the production system. And the change of the production system will imply an increase of the cost of the products in the supermarkets, in the market. Personally, I would be in favor of paying even 40–50% more meat. I find it quite unfair to the animals that we pay 1 kilogram of chicken as much as a bottle of Coke. But this is, of course, a personal opinion that goes beyond my expertise as a microbiologist and infectious disease specialist. I would like to close the presentation by acknowledging, of course, the European Union for supporting this important research program. And also, I forgot to say that part of the data that were presented were co-financed by the Independent Research Fund Denmark and by the umbrella organization of livestock production in Denmark, SEGES. And last but not least, you can see the faces of Christina, who performed, was the scientist responsible for the performance of the experiment, and Thomas, who was the supervisor. Thank you very much for your attention.